This year's prize is about how the optical microscope became a nanoscope. The Royal Swedish Academy of Sciences has decided to award the 2014 Nobel Prize in Chemistry to Dr. Erik Betzig at Howard Hughes Medical Institute, Ashburn, USA, Professor Stefan Hell at Max Planck Institute for Biophysical Chemistry, Göttingen, and the German Cancer Research Center, Heidelberg, Germany, and Professor William Murner at Stanford University, Stanford, USA, for the development of super-resolved fluorescence microscopy. Professor Sven Ledin will now give us a short summary, please.
Thank you, Stefan. Now, this year's could someone please do something about the resolution? That's a question that microscopists have asked for many years. Ever since the invention of the microscope, this has been the challenge. We see new things, but we see that there are more things to see. In 1873, Ernst Abbe proved that the theoretical limit to resolution is about half the wavelength of the light used. For visible light, that translates to about 200 nanometers. Now, a single human hair is about 100 micrometers. That's 500 times larger than this limit. So objects of that size are very easily studied with conventional microscopy. But most of the processes in chemistry and biochemistry take place at length scales that are much smaller. A typical bacterium is about 200 nanometers across, corresponding to the Abbe limit, which means that if we study bacteria in the light microscope, they will be featureless blobs, just like our laureates right now. This prize celebrates the circumvention of the Abbe condition. And 3 scientists have contributed to break this limit in 2 different ways. Stefan Hell developed stimulated emission depletion microscopy, STED. And Erik Betzig and W.E. Murner laid the foundations for single fluorophore microscopy. Now, why is this important? Electron microscopy has a much higher resolution and can be used to study structures in atomic resolution. But electron microscopy does not allow us to study living cells or the processes within them. The work of the laureates has made it possible to study molecular processes in real time. It's been used to study the dynamics of transcription and translation, that is, the reading of DNA and the transference from that to proteins. It's enabled us to look at how proteins associated with disease aggregate, for example, in Alzheimer's, Huntington's, and Parkinson's disease. And it's even shown us the structural dynamic changes to neurons in the brain that takes place during learning processes. Super-resolution microscopy doesn't only tell us where, but also when and how. And that is the greatness of this development. Biology has turned to chemistry. Chemistry has turned into biology. Guesswork has turned into hard facts, and obscurity has turned into clarity. Now, could we please have that clarity back? Lovely. Now, we can observe E. coli, just like the laureates on the screen, in all the glory of super resolution, without having to kill them, slice them, fixate them, and subject them to intense radiation and high vacuum. They can be studied in real time while they live long and prosper. Back to you, Stefan.
Thank you, Sven. Now I turn to Professor Måns Ehrenberg to give a guest presentation.
So this prize, as you have learned already, is about seeing. And what we have in front of us is a neuron, a nerve cell, and here to the left is the way it could be seen in the old days, before the laureates did their inventive work. And to the right, we see the neuron as it can be seen today, and it's labeled with fluorescence markers, and it's this fluorescence that makes the miracle possible. And another aspect of this, which was already mentioned, is that this is a snapshot, 5-second snapshot. So you can make a film of this and see how it grows and how it moves.